Let's talk about the energy of protein folding. So looking at this diagram, we have energy on our y-axis, remembering that high energy is unfavorable, low energy is favorable. So in this diagram, we can see a couple different things. First are that unfolded proteins are high energy. It's usually not very favorable for a protein to stay in that unfolded state. When I have a completely folded protein in its native state, notice that it's in a lower energy. And sometimes the proteins will kind of wiggle around a bit to do their job. So you can get, if you zoom into that, you see some conformational substates. But mostly what I want you to notice is that the folded protein is in what we call an energy well. So the idea is that it's at its local energy minimum. It's pretty happy there. It's prone to stay folded. As a protein is folding, it tends to get kind of partially folded into a certain folding intermediate. So maybe this alpha helix folds up, but this beta sheet isn't quite there. And that can be a somewhat stable conformation, but it's not the lowest energy minimum the protein can find. Or it could take a different path where maybe that beta sheet folds up, but the helix isn't quite there. So also notice in this diagram, we have this darker purple color here, which indicates normal protein folding pathways. So the protein starts unfolded and kind of falls down into these energy wells until it adopts its native folded state. So we also have some ways that proteins are not supposed to fold, or some kind of non-traditional folding structures. One example is if the protein doesn't fold at all, or if it's an intrinsically disordered protein, it stays up here. Another example are amyloid fibrils, which have a very low energy, very stable, but oftentimes not the correct protein fold. And another example are these amorphous aggregates, which is kind of like a mix of spaghetti. It's like a pile of spaghetti on your plate. Proteins contacting other proteins, and they're just kind of stuck together, but there's no nice, distinct fold to them. So how do proteins go from this unfolded state into this nice native folding state? Sometimes they do it on their own. Sometimes they need the assistance of another protein called a chaperone. So chaperones are proteins that help new proteins fold correctly. They do this by hydrolyzing ATP, so sometimes putting a little extra energy in, and also by providing a hydrophobic environment. So in this cartoon, we have a newly synthesized protein. It's like a little baby protein. It could hypothetically fold into the correct three-dimensional structure. Sometimes it can do that on its own just fine. The problem is sometimes this can be a slow process, and during that time, the protein may have a tendency to aggregate into like an amorphous structure, which is not correct, and then the cell would have to break it down and start all over again. So to prevent that, sometimes the cell will use a chaperone that keeps the protein in sort of a safe place while it folds, and then you have your folded protein ready to use. So the structure of these chaperone proteins are known, and they're really cool structures. They're giant, and they have this hole in the center of them. So that's where your new protein is going to go, where it's going to fold. And again, that chaperone can hydrolyze ATP, so put in energy to encourage the correct fold, and also can provide a hydrophobic environment in there to encourage correct protein folding. So what happens if proteins misfold? Why is that a bad thing? Well, one reason, we talked about amorphous aggregates, right? If you've ever worked with in vitro proteins in the lab, sometimes you'll have a protein solution and you'll start to notice white flakes, or the solution might become sort of opaque. That is when your protein crashes out of solution, is what it's sometimes called, which is when your nice soluble protein instead started forming amorphous aggregates. Once that happens, the protein is trash. You can't use any of that aggregated protein. So if you got some still in solution, you're okay, but this is all junk. When this happens in the cell has to just break it down back into amino acids and start over again. So that's sort of a waste of energy for the cell. Another example are amyloid fibrils. So this is the structure of an amyloid fibril, where the protein sort of gives up on all other intentional structures and instead just starts forming beta sheets. Lots and lots and lots of beta sheets. And these can be sometimes even millimeters in size, which is huge, absolutely huge, or maybe microns. You can see it with certain microscopes though. And amyloid fibrils can be toxic to the cell. So for example, the proteins amyloid beta, tau, and alpha-synuclein, they're all intrinsically disordered proteins, or they have portions of them that are intrinsically disordered. They all have the potential to misfold into amyloid fibrils. And amyloid beta is involved in Alzheimer's disease. Tau is involved in both Alzheimer's and frontotemporal dementia, and alpha-synuclein is involved in Parkinson's. So when these proteins misfold into these beta sheet-rich amyloid fibril structures that you see here, it can cause cell death, leading to those different neurodegenerative diseases. So then why would the cell bother having intrinsically disordered proteins if they can be so prone to misfolding, especially into amyloids? So intrinsically disordered proteins, or IDPs, can be really useful. And the reason why is because they change their shape in response to what is around them, which makes them excellent dynamic receptors. So for example, you can have a region of a protein that's intrinsically disordered that can act as a scavenger to store or neutralize small ligands. You can have, for example, a region of a protein that's intrinsically disordered that binds to other proteins to help assemble a larger protein complex. You can have an intrinsically disordered region that gets post-translational modifications, which we'll talk about that more. But that's like a signal to another protein that this enzyme is activated now, or it's deactivated. So there's definitely advantages to having these intrinsically disordered proteins in the cell. They have specific and useful functions. Okay, so we talked about protein folding. What about protein unfolding? So there's a word for that, and it's called protein denaturation, which is when a protein unfolds, meaning that it loses its quaternary, tertiary, and secondary structure. So a protein that's completely unfolded, we can also say that's a denatured protein. So common ways to denature a protein include urea, heat, and acidic conditions. So for example, if this is my nice natively folded protein, if I add urea, pretty high concentrations, 8 molar, and beta-mercaptoethanol, I can unfold the protein and break the disulfide bonds. So let's think about that. Why do urea, heat, and acid denature proteins? Well, for urea, let's start by thinking about the structure. What does this structure remind you of? Some parts of it look pretty similar to the carbonyl and the amine on the peptide's backbone. And we know those are very important for the hydrogen bonds that stabilize alpha helices and beta sheets. So urea competes for those backbone hydrogen bonds that make up the protein's secondary structure. If a carbonyl in the protein could just as easily hydrogen bond with the amine and urea, then it might do that, and then your alpha helix is going to fall apart. Heat. Heat disrupts non-covalent interactions through competing kinetic energy. So basically the idea is the more you heat things up, the more they're moving, the more and more kinetic energy you put in, and then that kinetic energy competes for the energy that would be gained through non-covalent interactions. What about acid? So to understand acid, we have to think back to what we learned about pKa values and how molecules can have different charges at different pHs. So acid has the potential to change the protonation state of some functional groups, which will redefine which interactions will be favorable. So proteins, most of them are optimized to fold at a neutral pH 7.4. If the pH starts dropping, some of those interactions aren't going to work anymore. Protein folding is theoretically reversible. So a denatured protein can be refolded if you, let's say you heated it up to denature it. If you slowly cool it back down, it should refold into the correct structure. Or if you used a denaturant like urea to unfold it, if you slowly remove that urea, you should be able to refold the protein. So the idea behind this, what's really cool, is that your protein should refold into the correct structure, and also that each protein has a unique structure. So then the implication is that all the information for protein folding is contained within the sequence. We do know chaperones can be helpful to fold some proteins, but for others, they can fold into the completely correct sequence all on their own. So what that tells us is that amino acid sequence is responsible for the protein's structure. So hypothetically, if you know the amino acid sequence of a protein, you could hypothetically predict how it's going to fold. Now, it's easier said than done. There's actually a lot of research on that. It's kind of hard to do, but hypothetically, that could be done. Okay, so intrinsically disordered proteins are particularly prone to misfolding into amyloids. Provide a hypothesis for why this is the case.